Bismillahirrahmanirrahim. Assalamu alaikum. Welcome to my YouTube channel, Science for Everyone. Today the video is related to RNA sequencing data analysis, and the title of my today's video is how to quantify your genes after alignment. As you all know that I have started a video series related to RNA sequencing data analysis, and this is the part 15th of that video series. In today's video, the concepts and steps you are going to learn is that what is gene quantification or feature counts, files required for gene quantification, performing gene quantification, understanding the Linux, the bash scripts, and understanding the practical and reshaping the scripts. So after watching this video, I am sure that you should have an idea about that what is gene quantification and why we perform gene quantification. And you are going to learn that what is the script that is used to quantify your genes after alignment. So in the part 14th of this video series, I show you that how to perform alignment. So after alignment, this step is very much important in order to quantify your genes, that how much genes are present in your specific sample. So this is very much important. Okay, so first of all, we should talk about gene quantification. Very much simple. Suppose these were your reads. These were your reads in the FASTQ files. Okay, I have explained that what are FASTQ files, and I have discussed a lot about the reads and sequencing. So I recommend to watch this playlist from the very beginning so that you could have an idea that what is RNA sequencing and how to analyze it. Okay, so for example, this is your read. These are your reads, and for example, this is gene C or any type of gene. Okay, so so if your 4 reads are aligned to this specific part of the genome, so very much simple, you should know that how much it is present. For example, if we count this, so these are four parts or 4 reads are aligned to your gene C. So then the software will decide that how much this gene C is present in your sample. Okay, so very much simple. This was the alignment. Okay, this was the alignment. But after alignment, we should know that how much a specific gene is present in our sample. Okay, so let's suppose this is gene B. This is gene B from here to here. So most of our reads align to this portion, to this portion, to this portion. So after that, observing this all alignments, at the last, the software, the tool that we are going to use today will decide that how much this gene B is present in a sample. Okay, so this is a very clear-cut concept. I will be not going to the algorithms that how it is doing it, but very much simple. If there is a gene B, there is a specific region of gene B in a reference genome, and your reads align to that portion of that gene. So then after that, as a whole, it will be decided that how much a gene B is expressed in a specific sample. Okay, so it's, I think, very much simple. Okay, so don't consider it gene A, just consider it gene C. No, this specific portion is not, this specific portion is only related to this gene A. Okay, so don't get confused. Okay, so this was a typical workflow. We perform quality control, then indexing, and this was that alignment. Okay, this was that alignment. We perform in the part 14 of this video series. So this was the script of the alignment. We use it, and after that we results, and these are that specific results. Okay, these are the log files. These are the log files, but this is the this part. This one is the output of the after the alignment. Okay, so these files consist of the entire information about the alignment. Okay, and this is this is the align.sorted by coordinate.out.bam. Okay, so this is the extension of it, and this was the sample. Okay, so for example, if we are having 2 samples of different IDs, so these are the 2 samples, and after that these are all the alignment information is present in this part. Okay, so very much simple. Let me tell you that when we perform alignment, so when these files are generated, so these files should be converted into another format. Okay, and that format is basically sorted.bam. Okay, first it was sorted by coordinate, and then we should sort it by BAM. Okay, so that it should be clearly and effectively processed by the feature counts algorithm. Okay, so for performing gene quantification, your input file should be in the extension of sorted.bam. Okay, so first the exact output was sorted by coordinate, and after that it should be sorted by name. Okay, so very much simple. There is a small code, a small script that very much simple. You have to just give these file as an input, and the software will process it, and it will convert this into sorted by name or sorted.bam. Okay, so this is a very simple concept. Okay, because these this concept is important for paired end files. Okay, if you are having paired end reads, so this should be important. Okay, remember. So this is the script of the sorting. Very much simple. Do not get afraid of it. These were the samples. Okay, these were the two samples of it, and then we have to specify the output directory. As I have mentioned in the previous videos, that what does this mean? So if you are dealing with these such type of files, so you have to change this portion of the you have to change this portion of the code. Okay, and it should be according to your own directory, according to your own PC path. Okay, so you can replace it. Okay, after that, do not change this part of the code, because it should be exactly the same. And then we apply a for loop. Okay, because we are having two samples, so we will apply a for loop. First, this part will be converted, this sample will be converted, then this sample will be converted. Okay, so that's why we perform for loop. Okay, so this all part is for debugging. It is important. If there is an error, it will show you. Okay, and I'm telling you that you only have to change this path. Okay, you only have to change this path. After that, we are converting align.sorted.by coordinate.output. So this will be the same, everything will be the same. You will have to just change the path, okay, of the script. I have run it and there was no error in it. Okay, so that's why. After that, the basic is this part. Okay, the most important part is this part, that we, and one point you have to remember, that same tools, okay, same tools are used to apply to process these files. Okay, so this is basically the package or the tool. Okay, so you have to install it. Okay, and I have shown you in the beginning of this video series that how to install a package, a tool into Linux. Okay, so I want you to just watch that. Okay, so same tools are used to sort BAM files. So very much simple, we call same tools, we applied 24 threads. Okay, and this is the entire path of it. Okay, this is the entire path of it. You have to change this, and after that, when it is changed, so then it will be sorted.bam. So very much easy, and this is also for debugging. After when it is successfully conducted, so then it will show you that yes, it is sorted. Okay, and if there is an error, so it will also tell you. Again, I'm telling you, only this part is the important part. Okay, and this for loop. Okay, all the other code is just for debugging, in order to run your sorting effectively with no errors and each and everything. Okay, so no worries, just apply this code and just change this path. Okay, according to your computer. So after sorting, when we perform sorting, so these files are generated. Okay, these were the inputs, these were the input files, and after that we got this output. Okay, very much simple. Now these files can be used for further processing. Okay, so very much simple. After that, when we get the sorted BAM files, these files. Okay, this here green color. Okay, these are the two files. Okay, so then these files will be used for the gene quantification. Okay, now we are not using these files. Okay, we are done with these files. Now we will be using this file 1 and 2. Very much simple. After that, this is the code of gene quantification. Very much simple. You have to change your this path, as I have told you. Okay, this path, it will be according to your own system. Okay, after that, this is the code. Okay, this is the code. You are not required to change this. This is the input, this is the output, this is the number of cores. So it should be arranged according to your computer. For example, if you are having core i7, so apply 5 cores. If you are having core i5, so apply 3 cores. Okay, 3 threads. Okay, all the other are the same. You only have to change this part. Okay, you only have to change this part. Okay, so there will be two types of inputs to perform feature count. Very much simple. The input files will be the sorted.bam files. One is this input. Okay, these are the files, and the other is the GTF file. This is the GTF file. As you have remembered that when we were processing indexing, so there was an annotation file. Okay, so this was the annotation file. I have explained the annotation file in detail in the previous videos, so I recommend to watch that video. Okay, so for gene quantification, you are required for sorted.bam file and the GTF file of the genome, of the reference genome. Okay, so you must have it, and you only have to give the path of it, that where is the GTF file located, where is the sorted.bam file located. And after that, once it is and you have to tell the output directory. Okay, that after that, when these processing is completed, so two types of files are generated. One is feature counts and the other is the log file, the summary. Okay, so this is the thing that we needed. Okay, so you have to tell it that where should the output files be stored. Okay, so it should be stored in this path. Okay, this one path. So this path was for the sorted.bam file. This part was for the GTF file. Okay, sorry, this was for the sorted.bam file. This part was for the GTF file, and this part is for the output files. Okay, so very much simple. Our output name will be featurecounts.txt. So we have to obtain this. Okay, and what is inside this file? I will show you that what is basically inside this file. Now, after that, when we when we get this feature counts.txt, so due to some reasons we have to modify it. Okay, we have to modify it. I will also show you in practically, but first of all you should have an idea that about the theory. Okay, so once the feature counts file is generated, so after that we have to process it. Okay, so just to clean it. Okay, because when it is I will show you that what is present inside this feature count.txt, but once it is generated, so there are additional columns that we do not need. Okay, so that's why we have to remove that additional columns. Okay, so very much simple. We will apply a code. We clean the unwanted column data from the feature count matrix. Gene id and gene expression related to sample should be included, and all the other columns should be removed. Okay, so column 1 is the gene id, while the remaining are the samples. Okay, so very much simple. We need column number 1, that is basically the gene id, and column 2, 3, 4, 5, 6, 2, 3, 4, 5, 6. These are the additional columns that we do not need. Okay, so when you are processing it, so these columns will be the same for all. Okay, so you can apply 2, 3, 4, 5, 6 to cut it. Okay, and the 7, 8, 9, 10, 11, 12, it depends upon your sample count. For example, you are having 10 samples. Okay, so 10 samples, so very much easy. One, the first column is the gene id, and the seventh column is sample number 1, sample 2, 3, 4, 5, 6, 7, 8, 9, 10. So if we are having, if we are having 2 samples, so very much simple, we will not include this. Okay, we will just include 7 and 8. So we will cut all the exceeding columns. Okay, that we do not need. Okay, so basically it was present in this feature.count.txt, so we will apply it, and after that, when once it is processed, so it should be feature counts modified. Okay, you are not supposed to change any of this, because this is the perfect code. Now this was all the theory. Now I will also show you practically that how it is performed. Okay, okay, so this is my Linux environment, RNA-Seq data, and I perform alignment. So after alignment, as I have shown you that the output is aligned or sorted by coordinate and aligned or sorted by coordinate. These are 2 files. Okay, I performed process alignment of 2 files, and after performing that sorting, so these files generated these. Sorted.bam and sorted.bam, and this is basically the location that I was talking about. So you have to change this location according to your PC or laptop. Okay, according to your Linux environment. And after that, we perform feature count. By performing feature count, I usually use these sorted.bam, and that GTF file of my reference genome. Okay, so you should have that in your folder. So after that, these counts were generated. These feature counts, and this was the summary. And when I perform modification, so it was modified. Now the main thing is that you can perform this practical all on your own. I have provided the scripts, the complete understanding I have provided to you. But let's see that what is the output. Okay, so let's see the feature counts, that how does it look like, and what does this code mean that I have explained it to you, that after the feature counts, we apply this code, and now then the feature counts are completely analyzed, and modified feature count matrix is generated. So first we will see the feature counts, then that is originally generated, and then I will also show you the modified portion. Okay, so this is my feature count. This feature count. So very much easily go to new and make a new Microsoft Excel file, and after that open this file. Okay, so this is an empty Excel file. After that, go to data, and from text you have to upload it. Okay, so where is my, this is the D portion, and this dealing, and this is RNA-Seq, and this was the matrix. Okay, so now we have the feature count, so I will import it, and this was next, and after that let me see. Okay, so next and finish. Okay, so it will take some time because it is a heavy file, and it cannot be easily viewed in Excel. So 76 MB of file need a little bit time. Okay, so just look here, it have a lot of information. We are having chromosome information, its start, end, this is the end. The other portion, we are also having the strand information, the length information, and the counts. Okay, so as I have told you that we do not need this portion, this column, and after that we also need this end and strand and start. So that's why I told you to remove these, so that you could get the modified file. Okay, so after that, how to modify it? So I told you that using Linux operating system, very much easily, you have to just remove you just have to include column number 1 and 7 and 8. These are basically your counts column. Okay, so it all depends upon your sample count. Okay, so the in between the 2, 3, 4, 5, 6, those columns are removed, as I have shown you that these columns are related to the end, start, strand, chromosome, etc. and etc. Okay, so let me show you that how does the modified file look like. So from text, and after that when I perform modification, and so this was the modified version. Okay, so very much easily upload it, and after that finish, and okay. So now it appears. So this is exactly the opposite of it. Okay, so I have removed those excessive columns. Okay, so this is the gene ID, and these are our samples. So if I applied it on 2 samples, so this will appear. Okay, so this is 10 samples, and I will be processing this matrix in RStudio in the later videos. But you should understand, in order to achieve this matrix, these all 14 previous 14 videos were all about it. Okay, so from raw sequencing we obtained this matrix, and then using this matrix we will analyze it and find our differentially expressed gene. Let me explain it briefly. This is our gene, and after that inside this file sample, this SRR156993854, inside this sample the count is 13. Inside this sample, the count is 11. And let me show you that what does this mean is basically we will refer to the metadata, and we know that this sample is related to a cancer and this sample is related to a normal. So we can then compare it using R. Okay, so we obtain this matrix. Basically, this is known as a gene matrix. Okay, in the rows, these are the genes. In the column, these are the samples, and we know that which sample is of cancer, which sample is of normal. So it depends upon your study. Okay, so these are the gene counts, and we will analyze it. Okay, so after that when you come across this gene matrix, so our Linux operating system data analysis is completed. Now we will use this gene matrix and this metadata file, and we'll process it through R using RStudio, and we will find the differentially expressed genes. Okay, so I think you have an idea that what was this all Linux data analysis about. We start from the very basics, and after that we obtain our gene matrix. Okay, so this was all about today's video. If you understand it, so like it, share it with your friends, and inshallah in the part 16 of this video series we will start with R programming, and we will analyze our gene matrix data using and metadata, and we will find the differentially expressed gene. We will also visualize our data to PCA plot, through volcano plots, through heat maps. I will also show you that how to generate it. Okay, so this was all for today. I will meet you in the part 16 of this video series, and subscribe to my channel and share it with your friends. Thank you very much. God bless you. Allah Hafiz.